So hello guys, welcome to my channel Dreamer Doc and this is the fourth video of the series Molecular Biochemistry. Till now we have covered up to DNA replication and transcription, and now today's topic is DNA RNA translation and Lac Operon. Last topic is Lac Operon in this molecular biochemistry. So you can see my earlier videos. There are total 3 videos in this series, and I will cover some more videos regarding the cellular biochemistry, rest of the metabolism topics, and all the subjects specific for INI-CET. The in-depth and concise video both I will make, and you should stay tuned for the more videos. And let's start with RNA translation right now. Start with the RNA translation. It is also very smart topic. You have to think, no, but that only two things, and it is all sorted. It occur in cytoplasm of ribosomes. Polypeptide chain is formed. RNA to protein is translation. That also we know. If misfolded proteins are formed, there is a diseases like prions. This is also we know. Na? Misfolded proteins are accumulated. That is an infection. But in hereditary, we will learn in further course. Okay, 70s is prokaryote is odd number, and 80s is eukaryote number. So they are large and small subunits. And Swedberg, sometimes they ask the basic question like what is S is sediment or Swedberg. Okay, size like is it mnemonic like size. So Swedberg. So antibiotic target 70s. So antibiotic we give like they target the 70s ribosomes. Okay, so next is child diagonal sequence. These are in prokaryotes, and it is an initiator sequence which is required by prokaryote. The initiator RNA required this child diagonal sequence in prokaryotes to carry on this translation process. Okay, in mRNA. And this Kozak sequence is in eukaryote. So prokaryote shine, and Kozak is K means eukaryotes. Okay, just remember that K is in both, but child diagonal shiny prokaryotes are shining. Okay, so remember this. Kozak is AUG in our methionine. Methionine is a starter codon we know, and this is in Kozak sequence of eukaryotes. So this can remember. Kozak is the key in eukaryotes and shine on start. So, and one thing also, this is a highly extra edge, guys. If you are aiming for under 500 rank or something ekdam clinical types, so this is, otherwise you are not required to learn in such a depth. In case I am going to tell you, you can remember or forget it. It's on your own. So in eukaryote initiation factor, what are that are used in translation? Total 6 are there. Okay, 1 and A are the scanning of mRNA is done by eukaryote initiation factor 1 and A1A. Okay, EIF to deliver the initiator RNA. We have to make this mRNA readable by this tRNA. Okay, this reads the mRNA. This joins with the ribosomes. And in eukaryotes, it is 80s, and in prokaryotes, it is 70s. So this mRNA should come in this, and tRNA should bring that mRNA to this ribosome to ribosome. They move from codon to codon. Okay, in ribosomes only. This is a E site. This is a P site. Oh, sorry. From left to right, this is acceptor side, this is peptide, this forms a chain. Okay, and this is the exit side, just remember like that. And this is in our mRNA. And this tRNA, this is a clover leaf shaped tRNA which has an anticodon loop, which has an acceptor loop that is ACC 3 end, and this is a D arm and this is a T omega arm. So D arm just reads the tRNA synthetase whichever is coming. Okay, and this attaches to ribosome. Okay, ribosome, and this actually detects which tRNA synthetase is coming, which tRNA synthetase is joining with this mRNA. Okay, and this joins with ribosome. Okay, ribosome. So these are all important aspects of this tRNA. Now this comes and this just goes like this. Acceptor side, exception is methionine. Okay, methionine directly comes, if there is a AUG sequence, this directly come to this P site. Okay, and now this comes, they form the bond from left to right, they exits and they move from whole ribosome. Take this and empty ribosome it left. Okay, they read the mRNA sequence. So this is whole crux, but you have to remember what is the defect. Okay, defect, because in diseases they ask like that only. This diagram, there is a mistake, this is anticodon loop and this should be T omega loop. Okay, so this and this a ACC bond is 3 end. Okay, so this is, or this mRNA is readed by this anticodon loop. It accept the amino acid and read in anticodon fashion, and the mRNA is has this binds with mRNA in anticodon sequence, take these two ribosomes, read in this direction and delivers the peptide bond. Okay, peptide bond. Okay, so, oh sorry, again this is acceptor, this is this is exit, this is peptide bond formation. Then they require tRNA synthetase, that is called charging. Okay, this is acceptor side, this incoming tRNA is read by this ribosomes and mRNA by this sequence. Okay, so I have told you in very concise way, you have to remember these things now. Okay, so in 1 2 I deliver the tRNA. Okay, it initiator tRNA is delivered by elongation factor 2. 3 reads the block the this RNA can go and bind to 40s. Now we have to remove this. We don't mind to now premature 40s, we don't want that association. So this 3 does that, 60s should come. Okay, 60s ribosome should come and bind to this initiator tRNA. Now E4 recruits like it has many function, I have written it here. Like it bind to mRNA cap, it scaffold protein is found. Basically this EIF4 does many things, just recruit the 40s to mRNA. No? EF3 was blocking earlier 40s, and it wanted to go with 60s, so EF4 is binding with 40s. EF5 hydrolyzes on EF2, this EIF2. Okay, this GTP initiate, I told you now this is extra, just remember what does they do. GTP, okay, this GTP was bonded by this, and EIF2, this helps this in hydrolyzing this. Okay, hydrolyzes EIF2. So the start codon recognition is helped by EIF5. Just remember that the GTP hydrolysis is important for the initiation. Okay, initiation factor. So prevents premature. Now we don't want to, again it goes and bind with the 60s which binded by 3. So 3 and 6 have same function. Like 3 is blocking premature RNA ribosome attachment 40s, and this is blocking the 60s. Okay, here it was giving to 60s, now it wants to bind with 40s. So this is the sequence of eukaryote, like 60 plus 40 thing, hai na? But just to forget this all, just remember if they are dysregulated then they can cause cancer or neurodevelopment disorder. If they are going to so much detail, then also they can ask this also. And if you are listening to my lecture, you will surely remember if anything comes like that. Okay, so don't over pressurize yourself, overburden yourself. So just remember A1 A2 scans the all mRNA sequence, and the EIF2 first deliver the initiator tRNA, 3 blocks premature 60s, 6 blocks the 40s. Okay, and now the EIF4 recruits the all thing. EIF4 has many parts like mRNA capping, scaffold protein forming, helicase binding, like everything. Okay, 40s. Basically, it's recruit to 40s. Okay, because we have blocked earlier 60s, now this is recruiting 4 say 40. So much easy now. GF hydrolyzes to GTP. This is the initiator step. Okay, tRNA synthetase plus GTP should combine. This is called charging process. Okay, so to start the translation. So 1 2 3 4 5 6 scan, deliver, block, recruit. Just see one more time and you are sorted. Dysregulation can cause cancer and neuro disorder. And child organo is for prokaryotes. Initiator sequence with align the ribosome with AUG, mRNA ka AUG sequence, and COSAC is for eukaryotes. Just remember that. And this is the process, like in very simple form I have written, like initiator, or that process, acceptor, yeah, EPA you can acceptor. This RNA comes from there, tRNA. This reads in mRNA in this direction. Peptide form bond is formed, exit, and empty ribosome is there. Okay, so it all happens in ribosome. In prokaryote, in eukaryote it is, and it is in prokaryote it is 70s, 40 plus 30. So this is so simple. Initiation, elongation, termination, 3 steps are there. Okay, in RNA also we have learned in our 12th 11th also that sigma factor is released and rho factor is released. Termination factors are also released in our sequence. Okay, in eukaryotes. Okay, so this is all the RNA thing. And now Lac Operon. Lac Operon is very simple. Just we have to just remember that to start this process we need lactose. Okay, we need lactose, and we need that glucose should not be there. Okay, glucose should not be there, then only Lac Operon will happen. E. coli inducible operon, it is an inducible operon. This is also asked. Okay, semi inducible, no, this is inducible. Just lactose metabolism controlled by that. Classic example of prokaryote regulation. So lac Z, what are they formed? Sometimes they ask what is gene ZYA. Just remember, Z makes beta galactosidase, Y makes permeases. Sometimes we get confused with Z and Y and A. Okay, so transacetylase. These are the enzyme required by E. coli for their metabolism of lactose. Okay, if there is a mutation in Z, no lactose breakdown, because beta galactosidase is required for this lactose breakdown. So if there is a mutation, no lactose breakdown. So three scenario we have. So first thing, you just learn the step. So first, this repressor is always binded to this site. Okay, repressor is always binded to operator site. So this is not happening, and always in repressed disease. But if suppose glucose is present and lactose is absent, so glucose is present, we don't need this. So now repressor is activated, repressor is always bind with operator, and this does not happen, this translation does not happen. But in case glucose is absent and lactose is present, this is favorable for our inducible operon. So this the just go away. Okay, repressor is not binded to operator. Now promoter will activate operator, and now this translation happens, and this cap and camp are activated by glucose. So this makes this camp and cap activation, and this activate repressor. This is also there. Okay, so cap and camp are activated by glucose, and we don't want this to get activated. If lactose is present, then only this repressor is kept away, and promoter operator goes on. Okay, so this is the simple thing. Three scenario are there. If it is absent, nothing can happen. Okay, no activation. If it is present, then it is fully operated. If at all glucose is present, now glucose is present but lactose is limited, okay, very less, so less enzymes are formed. Present but less enzymes are formed. Okay, so these are the three scenarios. Okay, small proteins are formed, less number of proteins are formed, is lactose is small amount. Now glucose is also present, then small, little expressed. So these three scenario are there. And questions, I have not seen like very important, very high weightage questions, but okay, like um, it's okay, we have to learn that. Okay, it's clinically also important. So that's it, guys. And one more thing I have to say that is frequently asked from our translation thing is not the steps but post translational modification. They mix some four options and they say which is not translation, which is there, or assertion reason thing they can ask. So the post translation modifications, you should learn every name. Okay, just name: phosphorylation, glycosylation, anything which is added like hydroxylation, methylation, acetylation, and ubiquitization. That is folding of the proteins by ubiquitin. Okay, ubiquitin proteasome pathway which breaks the protein sequence. So ubiquitation, these are all post transcriptional. Okay, just read this by heart. No need, if you are reading by heart every step, everything, no, there is no need. Okay, just if you are not understanding, if you are not able to tick, then your all this knowledge goes in vain. Okay, this happens every year with every topper, every aspirants. They are not able to recall this. They can say influently like this viva thing also every time, but they are not able to recall this. So what's the point? Everything is judged by your exam taking skill now. So right now, so you have to learn this. Okay, phosphorylation, glycosylation, hydroxylation, methylation, acetylation, ubiquitation. They are post translational, not transcriptional. Okay, so just remember this. And chaperone protein is something like protein folding protein hai. HSP60 they are called, heat shock protein. So they help in yeast cell, they make the folding of protein. Okay, they are intracellular. So in high temperature they are expressed. Okay, high temperature, pH, acidic pH, hypoxia, so they help to protein, means they are uh, able to make the protein folded. Okay, and they help that yeast should not be, it means the protein should be not misfolded. Okay, not misfolded or protein denatured. So these help in, because in high temperature, high pH, protein is ready to get have tendency to get denatured now, so this help. Heat short protein are called chaperone protein. Okay, chaperone, this is also favorite for AIIMS. Okay, the chaperone protein, they facilitate the protein shape. Okay, so these are the thing. And ubiquitation is the proteasome pathway is like tagging the proteins to lysosome so that they get degraded. Okay, that misfolded protein, these are formed in anyhow. If this process is not going on smoothly, so this misfolded protein were later tracked by this ubiquitation, and this comes under this post translation modification. So this is very important thing. Don't confuse with the chaperone protein and the ubiquitation, because they both are they both comes in options every time. Okay, so just understand what is that. Okay, I had told you. So that is the most important thing in there. And I think I have told you everything regarding this. And this is a must know. Okay, if you know this, then your molecular biochemistry DNA RNA part is simplified, and you are sorted for exams. Okay, if you know these basic things. And the rest topic in biochemistry are like cellular pathway and metabolism pathway, and like genetics pathway. Okay, the inheritance, X-linked dominant, and this pedigree chart and all, and cellular pathway, cell structure and all, I will cover in the next classes, I think. And if you have any doubt regarding this, and anything regarding this, you can surely comment this, and stay tuned for more videos. I will simultaneously teach every topic from whole 19 subjects in detailed integrated manner, very INI-CET specific, and especially NEET PG specific also. Okay, so thank you for listening, guys. Have a great day. Okay.